So this is Dr. Matt Wimmer from Temple University. So he wants us to introduce him by beginning with this thought here. The once controversial idea that parental experiences such as stress or diet can shape the physiology and behavior of their offspring via epigenetic mechanisms has become an active area of research. Dr. Matt Wimmer studies the influence of drug abuse in fathers, sires, on future generations. His research program combines animal models of drug addiction and memory formation with molecular biology techniques to investigate the impact of paternal drug taking on drug-related behaviors and memory formation in progeny. Dr. Wimmer is also interested in epigenetic remodeling events in the brain that underlie these inherited changes in behavior. Dr. Wimmer received his PhD from the University of Pennsylvania under the mentorship of Dr. Ted Abel. His postdoctoral training under the guidance of Dr. Chris Pierce at Penn focused on the transgenerational epigenetics of cocaine addiction. Dr. Wimmer's research is funded by the National Institute on Drug Abuse.
Thank you, Jake, for that very kind introduction. And I'm so grateful to have the opportunity to share some of our work with you today. And I want to especially thank the Penn NGG students and all of the organizers of the Neuroscience Public Lecture for inviting me. And so, yeah, the title of my talk is The Apple Never Falls Far. So I hope if I do an okay job, you'll understand what I mean by that by the end of the talk. And I'm going to talk to you about using multi-generational animal models to unravel the mechanisms underlying addiction susceptibility. So we all know that there's an ongoing opioid crisis, and it was at least in part driven by a sharp increase in the prescription of opioids. And so in the past recent years, in any given year, about 65 million individuals will be prescribed opioids and exposed to opioids. But what many of you may not realize is it's actually a relatively small proportion of individuals for whom this becomes a problem and they go on to actually abuse the drug. And so as a scientific community, for several decades now, we've been focused on trying to understand what's different about these individuals. And can we find reliable ways to identify them before they get exposed to the opioids? Because that would have a profound impact in the clinic for managing pain and deciding who should and shouldn't be prescribed opioids. And so this idea is really going to be the central focus of my talk for today. And substance use disorder is a chronic relapsing disease. It's really difficult to manage because it has long-lasting consequences long after the drug is no longer on board. In this diagram here, I don't know if you're able to see my cursor, but in this diagram, I'm showing you sort of the progress of the disease with this initial exposure. For example, prescription of oxycodone after surgery or after an injury, that for that vulnerable subpopulation can turn into this chronic use, followed by periods of abstinence. And unfortunately, the current relapse rates, the number of individuals who return to that chronic compulsive use after the periods of abstinence, is currently very high. So from a public health perspective, there are at least two areas of potential intervention. One is the relapse and trying to keep substance use disorder patients abstinent through treatment. I'm not going to talk a lot about that today, but I did want to highlight the fact that particularly for opioid use disorders, there are currently available and efficacious treatments. And so if we really want to help a lot of people quickly right now, one of the things we should focus on is to give those individuals better access to the treatments that work. I also believe that we need to develop new therapeutics. And in fact, my lab and many others are really focused on that. But that's going to take a little longer. And so, better access could have a larger impact quicker. And the other area of intervention that I am going to focus on today is prevention. And that gets back to this idea of trying to identify the vulnerable subpopulation in part. And I think there's a lot of interest in these prevention efforts, because in the long term, we can really save ourselves a lot of trouble. Once individuals fall into this vicious cycle of addiction, other problems are likely to emerge, both resulting from chronic drug use, but they also may contribute and perpetuate and make the relapse more likely. So if we can stop this from happening in the first place, that also has a lot of value from a public health standpoint. And so that really was the question that drove the research that I'm going to share with you today. We wanted to try to identify the molecular mechanisms that would be predictive of addiction susceptibility. And we're doing this using some novel and a little different approaches that I'm going to tell you about. So I'm really lucky that I'm going last here, because the previous speakers have done such a great job of introducing and explaining what epigenetics are. And we know going into this that substance use disorders are heritable, meaning they're inherited from mom and dad, and they're passed down from generations to generations. We've heard about DNA. We know that for substance use disorders, about 40 to 70% of the risk to develop substance use disorders can be explained by genetics. But what about the rest? Well, you know where I'm going. We've heard a lot about how environmental factors encoded by these epigenetic mechanisms are so important. And so this really comes to a chicken or an egg problem. If parents who live with substance use disorders have children who are more likely to develop psychiatric illnesses, is it because of the genetics, or is it because of their own drug use and their environmental influences during their lifetime? Well, I'm not going to provide the answer today, but I can tell you that probably the answer is it's a complex interaction of the two. But what I'm going to focus on is, of course, the epigenetic component. And one of the reasons I wanted to pursue that in my own lab is that it's an area that's really understudied and really difficult to address historically. So that brings me now, we've heard a lot about the brain, and I'm going to get back to the brain by the end of my talk. But I want to take a little detour and tell you about how these epigenetic processes, they're involved in all tissues, and they're really important, including the germline. And so this idea of epigenetic inheritance was first really posited by Jean-Baptiste Lamarck in the 1700s. This idea of acquired traits, he's really famous for this example of the giraffes. If a giraffe is stretching its neck to reach higher leaves, the idea is it could then transfer that experience to the next generation of giraffes. And so over a very short period of time, the giraffes could have acquired their long necks through stretching that's transferred across generation. At the time, the scientific community completely rejected this idea. But I'm here to tell you that the last 3 decades or so suggest that maybe he wasn't completely wrong. And both in human studies as well as animal studies, there's actually very strong evidence to suggest that parental life experiences can influence the biology of the next generation via this epigenetic inheritance and these epigenetic mechanisms in the germline. And we really leveraged that literature when we try to develop our animal models of substance abuse across generations, these multi-generational models of substance abuse. And the basic idea, the basic premise really was to say, what if the insult in the parent generation, in this case, I'm going to tell you about morphine exposure, conferred the vulnerability or part of the vulnerability in the next generation when they become adults. So all of the much of the work that I'm going to show you today was spearheaded by Dr. Andre Toussaint, who was a PhD student at my lab and recently defended his thesis. So some of you may be wondering, well, why not do this work in humans? And so here I simply wanted to acknowledge the fact that a lot of this work is ongoing these genome-wide association studies or GWAS studies in humans are really trying to find that genetic link between substance use disorders or other disorders and the genes that are responsible for this. I also wanted to highlight, though, that these studies are extremely expensive and logistically very difficult. And so while it's critical that we get this information, it's also been a long haul to try to get these answers. And in fact, as far as opioid use disorders is concerned, those studies have just been published within the past couple of years. And so one of the reasons we chose to focus on animal models is because they really allow us to kind of pare down the question to a more simple form. And we can also hold some of these factors that increase the complexity in human populations steady in the lab so that we can really focus on just one factor or another. And in our case, we're really focusing on the drug exposure and the parental generation. The idea there, by using these simpler systems, is to generate some targets in rats or mice. And then our work ultimately is to then translate those back to the humans and figure out whether or not they're relevant in the human population. But you know, historically in neuroscience and biology in general, there are many examples where this has worked. A lot of what we've learned has been from flies, mice, and rats. So there is homology, and that's what we're hoping to use in our studies. So here's how the experiments work in my lab. The fathers or sires self-administer morphine for 60 days. So they're in these boxes, and every time they hit a lever, they get an infusion of morphine. This IVSA or intravenous self-administration model, I really like because it mirrors a lot of the conditions of human drug consumption and human substance abuse. And the reason we gave them access for 60 days is that is the duration, that's how long it takes for a sperm cell to be produced and mature all the way to be able to fertilize an egg. So we wanted to capture the entirety of the lifespan of a sperm cell, if you will, and we wanted to capture that exposure to morphine during the entire time. At the end of the 60 days, the animals are then paired with a female that's never gotten drug before. And everything I'm going to tell you about today is in this first generation or F1 generation here in purple and green. For our control, as opposed to morphine exposure, the dads only got saline, right? So if you follow the morphine arrow, the morphine sired offspring were produced by a morphine exposed sire or dad, and the saline sired side, their dad only got saline. Now, in the green, I'm showing you the males. In purple, I'm showing you the females. I'm unfortunately not going to have time to tell you much about the females today, but I'd be happy to talk to you about it in the Q&A. But I wanted to highlight that it's really important to study both, in my opinion. So the first question we asked is if we take our morphine-sired progeny and we give them access to those morphine self-administration boxes, what happens? And in the dark green here on this graph, you can see that what happens is they take a lot more morphine than the controls. And this brings up another question, because they could be taking more morphine because it feels better, right? Because the reinforcing efficacy of the drug is higher. Or I could argue that they are consuming more morphine because they need more to feel the effects. So to get at that question, we did another experiment. In the first part, on the drug taking, every time the animal hits the lever once, they get one infusion of morphine. In the motivation side of the graph, we changed the rules. The first infusion costs 1 lever press, but the next infusion is going to cost 2 lever presses, then 4, then 8, and so on and so forth. And so throughout that session, the animals have to work harder and harder to earn a single infusion of morphine. So we're really getting at the motivation to earn these morphine infusions. And what you can see is that if dad took morphine prior to conception, his sons actually work a lot harder. And what that tells us then is that it is the reinforcing efficacy. Morphine just feels better for that population. They're more sensitive to the euphoric properties of morphine, if you will. So what about pain, right? Anytime you talk about opioids, those are the two sides of the coin. And so to address that question, we collaborated with Dr. Ismail Abdul-Sabur, who's at Columbia University, and Dr. Nate Freed. And I really needed them because they're the expert in pain neurobiology. And so Nate and Ishmael had recently developed a novel pain scale in mice that we together translated to the rat. And so those two graphs here are showing you that our new pain scale is able to differentiate touch-like stimuli, like with a cotton swab or with a paintbrush, compared to a painful stimulus like a pinprick, right? And so armed with, and I don't have time to go into much more detail about that, but I'd be happy to answer questions. But armed with this new tool, then, we were able to ask about our morphine-sired generation, whether or not the pain-killing properties of morphine were different in those animals. And surprisingly, the answer was yes. So they were also more sensitive to the antinociceptive properties, the pain-killing properties of morphine, when we used the novel pain scale. So if you believe everything I've told you, when dads are chronically exposed to morphine, they're producing these vulnerable generation of rats, morphine-sired rats, that are more sensitive to morphine, both in terms of its addictive properties and also in terms of its pain-relieving effects. And of course, if this were, as I said, if we were able to show this is true in humans too, well, that could have an immediate impact in how we think about pain management and prescribing opioids. But beyond that, I think in my own lab, we now have a very unique opportunity to get back into the brain. I know this is a neuroepigenetics talk, so I'm finally getting to it. And so now, if you believe me that these animals are more sensitive to morphine, now we can look in the brain, in the reward pathway, in the centers that influence pain, and ask about what's different about these animals. We can use gene expression assays that Erica mentioned. We could look at the neuroepigenetics that you heard from Han-Jun Son. And the idea here is with these genome-wide screens in this vulnerable population, we can come up with new targets that we can hopefully functionally validate later on. So I'll just tell you very quickly a little bit about the reward pathway, where the ventral tegmental area projects to the nucleus accumbens, and the VT, both areas have these mu opioid receptors, where morphine has its main site of action. So we asked about those receptors in both brain regions in our F1 population. And what we find is there were no changes in the accumbens, in the NAC, but in the VTA, we see an increase in mu opioid receptor binding. And that could potentially explain why morphine was more reinforcing in those animals, for example. In another experiment, we took all three brain regions, and I'm adding the periaqueductal gray, which is a brain region that's important for pain. And we did the RNA sequencing that Erica talked to you about to look at what genes are differentially expressed in each of these brain regions when we compare morphine-sired to saline-sired animals. So in the volcano plot on the left there, in the green, in the blue and yellow, are some genes that are differentially expressed in the periaqueductal gray. They're part of what's called the RGS family of proteins that we know influence opioid signaling. So there's another potential target, this time in the context of pain, that may be accounting for a higher sensitivity to the pain-killing properties of opioids. And in the discs, I'm showing you the number of genes that are changed by paternal morphine exposure. And what you might notice is that there's very little overlap between the discs. So what that means then is that paternal morphine exposure produces unique reprogramming events in each of these brain regions. They each have their own signature. And so we have unique set of targets in each of these brain regions that could be accounting for some of the changes in behavior I showed you in the previous data. And our job now is to go back and try to validate some of them, to find these new targets, to both predict and maybe even treat eventually substance use disorder vulnerability. And I will leave you today with this idea that your tax dollars are very hard at work. This is about 5 or 6 years of work I've just shown you from a very talented team of individuals, postdoc, graduate students, undergraduates. And I just feel so fortunate to have been able to work with them, and I wanted to acknowledge them here. And I also wanted to acknowledge all of our collaborators and, of course, our funding sources. NIDA, the National Institute on Drug Abuse, funds most of our work. And last but not least, I want to thank you for taking time out of your Friday to come and listen to us today, and I'd be happy to try to take any questions. Thank you.
Hi, everyone. Great talk, Matt. I was just going to ask, because I'm assuming obviously you used rats, if you have a favorite rat. But we do have a question from Ethan. He asked, have you tested the vulnerability of morphine-sired rats to other drugs? If so, how specific is this effect?
Yeah, thanks for that question. So we've tried cocaine self-administration and didn't see any differences. I should also note, actually, that the changes I showed you for morphine self-administration were dose-dependent. So at lower doses, the animals were more sensitive, but if we increased the dose of morphine, we saw the difference went away. And it was specific to opioids as far as we can tell. Cocaine, there was no difference. Nicotine, there was no difference. And the animals were also pressed for sucrose pellets, which we consider to be a natural reward, and that was also not changed.
Ashley asked, have there been efforts to utilize machine learning models to predict drug abuse vulnerability from genome-wide DNA methylation patterns in mice or humans?
So that might be a better question for Han Jun Song, but I think there are genome-wide screens for methylation. As I said in the beginning, I think the GWAS is just now coming out, so I don't think, at least to my knowledge, there hasn't been any of those experiments done yet. At least not in the brain. What I can tell you, I know of a few studies at Duke where they actually looked at the germline of patients with substance use disorder to see if they could find patterns in the germ cells and the sperm cells, and they looked at methylation there. But I don't think any of those studies have been published yet. I think I've just seen them at conferences so far. But that's a great question and a great idea.
Definitely. All right, I did see the question. Okay, there we go. Jonathan Rimley asked, do you know how many generations after opioid exposure it takes for offspring to return to a similar level of opioid susceptibility as an untreated mice?
Yeah, great question. So if I'm understanding what you're asking correctly, in the first generation, we see this phenotype, right? We see this change in drug taking. Do the grand offspring also have the same problem? Or does it go away? Or how many generations does it take to wash out? I think that's a great question. And it's something we're interested in, but we haven't had the time to do it yet. What I can, however, tell you, which is somewhat related, is everything I've shown you, we capture, if you will, we capture the exposure right after. We made the animals right after they're done self-administering. And so, and that's what I've shown you. But what I didn't have time to show you is we can wait 3 months, right? We can wait 2 rounds of spermatogenesis where all of the sperm cells should be relatively new and have been developed without the presence of morphine. And when we do that, if we wait 90 days before we breed them, many of those phenotypes actually go away. So I don't know the answer about future generations, but I can tell you that within the parental generation, it doesn't seem to be a very long-lasting effect. If you wait long enough, it seems like that the experiential information does not get passed on to the next generation to confer the vulnerability.
I have a question because you briefly touched on just males. Do you mind sharing maybe like 2 little points what you found in females? Because
Yes.
aren't more females more susceptible, or we're seeing a huge opioid crisis, I feel like more in females, or yes, I don't want to go on a tangent, but go ahead.
Well, it's interesting. So in the females, we actually didn't have a drug-taking phenotype, and we were very surprised by that. On the other hand, we've heard a little bit about object recognition and these object-based tasks, which we also used to determine whether cognition would be affected. And that particular measure was only affected in females. So many of the things I've shown you today do seem to be sex-specific, meaning it's either the sons or the daughters that seem to be impacted. And I think that raises a whole nother set of questions about why there's sex specificity. And you have to remember there are so many developmental steps between, the insult and when we're looking in the adult offspring that we're starting to think about it, but we really need help from developmental biologists to weigh in and help us with that. But thanks for that question.
Okay. We also got a question from our very own Jake. Why would an epigenetically driven increase in opioid sensitivity be evolutionary desirable? If the father is in an opioid-rich environment, wouldn't it make evolutionary sense to downregulate opioid sensitivity in offspring? Ooh.
That's a great point. As I like to tell my lab, the data is the data. But you're right that, and in fact, we were surprised to find this. And in my postdoctoral work with Chris Pierce, we actually developed similar models, but with cocaine instead of morphine. And if the dads have cocaine, it actually does confer a resilience phenotype in the next generation. So I think you bring up really an excellent point, Jake, which is that and then maybe I oversold this a little bit in my talk. We have to be careful about, whether and how much this would apply to humans, because as I said, the rats aren't nearly as genetically diverse. They are rats and not humans after all. And so I think more about these studies as a way to delve further into the biology and really try to better understand what some of the mechanisms could be, rather than it being a comment on evolutionary biology, because I think you're right. And I think, in fact, in humans, there's evidence to suggest that it would go the other way, whether you're talking about stress or diet or other insults in the parental generation. So that's a very good point, and really highlights the complexity of these models in the humans.
Okay, we have a question from Joy. Do you have plans to look at epigenetic? Oh, I thought it was Joy coming on. Do you have plans to look at epigenetic marks in the morphine sire's sperm to see what is being transmitted to progeny to cause these effects? Ooh.
Yes. So I am not a developmental biologist, but I have talked to many of them and recruited some as collaborators, and work from Tracy Bale, actually, who was at Penn and now moved to Baltimore to the University of Maryland. Her work really highlighted that stress in the paternal generation can reprogram the small RNA content of sperm very quickly, and that those small RNAs are functionally relevant for conferring the phenotype to the next generation as far as stress, the HBA axis and stress is concerned. And there's also work from Ali Rando really highlighting how those small RNAs at early developmental stages can really influence the developmental trajectory of the next generation. And so we're taking our cues from that literature and thinking about whether or not we can define the small RNA signature of the sperm of our sires. And again, I think one of the reasons, DNA methylation, and there's some evidence that perhaps histone marks that Erica was talking about are also important in the germline. But I like the idea of going after the small RNAs first because of those really elegant potential functional experiments you can do where you can have in vitro fertilization and recapitulate the small RNA signature of the insulted parent and really delineate whether that is what's conferring, the changes in vulnerability or pain sensation, et cetera. Great question.
Okay, I think that's all the questions for you as of right now. I think we might move into our panel now.